Okay, we are going to cover our last chapter for Unit 2, the muscular system. So just like the skeletal system, make sure you pay attention as I am speaking throughout the lecture, and I'm going to highlight the stuff that you definitely need to know for the test and some of the stuff that you should not spend time studying for the exam. So these should look familiar back from the tissues in chapter 4 of unit 1. But we have three different types of muscle tissue. So we have skeletal tissue, which is attached to the bones. We have striated skeletal, so that means it's going to be striped. So that means that we have our actin and myosin, our two myofilaments that we'll talk about shortly. They create a striped appearance. So that's what striated means. And our skeletal muscle is our only muscle that's under voluntary control. So if you see voluntary control as one of your options, the only thing you can pick is skeletal. So striated and voluntary control is what you want to remember. Our cardiac muscle is called cardiac muscle because it's only located in the heart. It too is striated or has that striped appearance because of the two different myofilaments. But our cardiac muscle is involuntary. So we do not consciously think about our heart beating. Even if we focus, we can't make our heart beat. It is under involuntary control. And then our final type is smooth muscle. So smooth is called smooth muscle because it's non-striated. So under a microscope, it does not show up as striped. It does not have the two different types of myofilaments. So if you saw non-striated, the only one it could be is smooth muscle. This is also under involuntary control. And so this is the muscle that we find in our blood vessels and other hollow organs such as the bladder and the stomach. So it allows for some of our organs and vessels to function, but we do not control our smooth muscle. All right, just like our last couple systems, we'll start with the functions. Of course, the primary function for our muscular system is movement. It also helps us maintain posture. So as you're sitting or standing, even though we don't necessarily think about doing anything with our muscles, we have at least a little bit of muscle activation, which helps us maintain posture. We call this activation muscle tone. So your muscle tone at rest gives you your posture. So the only way that you're still staying upright is from muscle action, even though we're not consciously thinking about making our muscles do anything. We have a few muscles that help with respiration or breathing. We'll touch on those later on in the chapter. Production of body heat is one that you don't think about often, but our muscles generate heat as they work. So when you exercise, you heat up, and then think of even like something like shivering or your jaw chattering. That's muscle action that's trying to warm you up when your body temperature is dropping. The muscles of the face are going to help us with communication, constriction of organs and vessels. So as we said, like stuff like our stomach, our bladder, our blood vessels, they're going to have muscle in them so that they can actually move and function. So our stomach has to be able to churn up our food. Our bladder has to be able to squeeze out the urine, and our blood vessels have to be able to change diameter. And then, of course, cardiac muscle is responsible for contraction of the heart. All right, we're going to touch on a few functional properties of muscles. So you should know these for the exam. So contractility is the ability of a muscle to shorten forcefully or contract. So that should be an easy one to remember. Contractility is the ability of a muscle to contract. So excitability is the capacity of muscle to respond to a stimulus. So this is how well a muscle will respond to input from our nervous system. Extensibility and elasticity go hand in hand a little bit. So extensibility is the ability of our muscle tissue to stretch beyond normal resting length and still be able to contract. We'll touch on this when we kind of zoom all the way into our sarcomere later on in the lecture, but more or less, if our muscle gets stretched too much at some point, it will not be able to contract any longer. So extensibility is kind of the extent past normal resting length that the muscle can still be functional. So if it goes too far past that resting length, then it's no longer going to be able to contract. And then finally, we have elasticity. And so this is the ability of the muscle to recoil to that original resting length after it has been stretched. So imagine that we're testing the extensibility of a muscle. So we stretch it all the way to its maximum. If it easily and quickly is able to bounce back to its original resting length, then that would mean it is an elastic muscle. So elasticity is the ability to return from that stretched position. So extensibility is the ability to contract from a rest or from a stretched position. Elasticity is the ability to return to original resting length. So our skeletal muscle takes up quite a bit of weight when you add the connective tissue that's associated with it. About 40% of our total body weight is made up of skeletal muscle and its connective tissue. So of course our skeletal muscle is called skeletal muscle because it is what is attached to our bones and helps us move our skeletal system. So we already talked about skeletal muscle being striated. So it has that striped appearance under the microscope. And so our skeletal muscles are not only just muscle tissue, but they also include the nerve that supplies that muscle and the connective tissue that attaches it to the bone and helps it maintain its spot in the body. So during this lecture, we're going to do a little bit of zooming in and zooming out in terms of what makes up a muscle. So we're going to start that with our connective tissue coverings, and I'll go through and highlight these again as we go through the different parts of a muscle. So each skeletal muscle, the entire muscle, the whole muscle, is surrounded by a connective tissue sheath known as the epimysium. So the epimysium is tissue that surrounds the entire muscle. Within a specific skeletal muscle, we're going to have subdivisions into groups of muscle cells. And so these groups of muscle cells are going to be named fascicles. So an entire muscle or the whole muscle is made of bundles of fascicles. So each fascicle is surrounded and organized, kind of held together by a covering known as our perimysium. And then within our fascicles, we have muscle cells known as muscle fibers. And each individual muscle fiber is going to be surrounded by an endomysium. So we kind of went zoomed out to zoomed in here. So let's go backwards now. So our skeletal muscle cells are called a muscle fiber. They're surrounded by a tissue covering, which we call our endomysium. So think endo means inside. So endomysium is all the way deep in the muscle. So each individual muscle fiber is surrounded by an endomysium. Our muscle fibers are bundled together to create a fascicle, and our fascicles are surrounded by a perimysium. Groups of fascicles create a skeletal muscle, and the entire skeletal muscle, one individual muscle, is going to be surrounded by the epimysium. So a muscle fiber is quite a large cell. It's one of the few types of cells that's going to have multiple nuclei. So some muscle cells have several hundred nuclei spread throughout the cell. So we have quite a wide variety of size in terms of our muscle fibers. So they can be as small as 1 millimeter and as large as 30 centimeters, which is quite large. So we said striated gives a striped appearance. So this is due to the two different types of myofilaments, which we'll talk about a little bit farther along. And so after birth, our total number of muscle fibers actually remains constant. So growth of muscles is really just an increase in size of our muscle fibers rather than actually adding a higher number or an increase in muscle fibers. For our muscles to activate, they have to be activated by our nervous system. And for that to happen, it takes some electrical activity. So a couple of structures related to our muscle activation are the sarcolemma. So that is our cell membrane of a muscle cell is termed the sarcolemma. This sarcolemma of our muscle fiber or the cell membrane has a lot of inward folds that are kind of tube shaped or tube like, and we call these transverse tubules. So this is how the cell membrane is able to penetrate to the interior of our muscle fiber. So the T tubules are kind of associated or run near and large portions of our smooth endoplasmic reticulum or smooth ER. In the muscle, we call this the sarcoplasmic reticulum or the SR. Excuse me. So the enlarged portions of our sarcoplasmic reticulum are the terminal cisternae. So that's those enlarged portions of the smooth SR that are running near the T tubule. So two terminal cisternae and one T tubule is known as a muscle triad. So that's our electrical component or how we get that electrical signal to the center of our muscle fiber is our muscle triad. So our sarcolemma forms the T tubule, and then we have our SR, which has a high concentration of calcium. Calcium is very important for muscle contraction, and you'll see why as we go farther along. The cytoplasm of our muscle fiber is called the sarcoplasm, which contains many bundles of different protein filaments. So sarcoplasm, anytime you see sarco, it's going to be muscle. So you can see the trend. Our sarcolemma is the cell membrane for our muscle. The sarcoplasm is the the cytoplasm for our muscle fiber. So this shows kind of overview of the structure of our skeletal muscle. So I'm going to zoom in here just a little bit. So we're going to go from zoomed out to zoomed in first, and then we'll go back the other way. So we have our tendon that connects muscle to bone as a quick reminder. So our entire muscle is surrounded by an epimysium. That whole muscle is composed of different muscle fascicles. So these muscle fascicles are surrounded by the perimysium. One individual muscle fascicle is made up of multiple muscle fibers, and this is surrounded by an endomysium. So now we're going to go even more zoomed in than we have thus far. Our muscle fibers are made up of myofibrils, and our myofibrils are made up of myofilaments. So these are kind of our microscopic proteins. We have actin, which is the thin myofilament, and we have myosin, which is our thick myofilament. So now we're all the way zoomed in. We're all the way down to the myofilaments. So now we're going to go back up. So our myofilaments, a bunch of myofilaments combined to create a myofibril. Multiple myofibrils are surrounded by an endomysium, and that's what creates a muscle fiber. Muscle fibers are grouped together and surrounded by the perimysium, and that's what forms a muscle fascicle. And then our muscle fascicles are grouped and surrounded by the epimysium, which is what forms a whole muscle. Looking at the right side of our slide now, this is showing the zoom in on a muscle fiber. So we're here at this level. So you can ignore A band and I band. So the whole thing is a muscle fiber, which is surrounded by the endomysium. So you can see our cell membrane here is labeled as the sarcolemma. The cytoplasm of our muscle fiber is labeled sarcoplasm. One myofibril is this group here. So in this muscle fiber, we see 4/5, kind of 4.5 myofibrils. This yellow portion is the smooth ER of our muscle fiber. That we term the sarcoplasmic reticulum. Our sarcoplasmic reticulum ends in a terminal cisterna on either side here. And this T-tubule is an infolding of our sarcolemma. So those together form our muscle triad. So there's not a ton of questions on this kind of microscopic view of our muscle fiber and whole muscle, but to understand how the entire system works, we have to kind of go through this zoomed in view. So you should at least understand kind of the order of operations here in terms of zoomed out to zoomed in and vice versa, but don't get too caught up in labeling every single thing. Excuse me. So in your chapter 7 lab, you will do some labeling that looks relatively similar to this. So that's a good chance to practice and maybe take a screenshot or picture to review so that you can try to kind of help get this down. All right, I'm going to zoom back out here. Okay, our mechanical components. So we said our bundles of protein filaments, our actin and myosin bundled together are going to create a myofibril. So our actin is our thin filament, our myosin is our thick filament. So actin and myosin are arranged into this special structure that repeats itself known as a sarcomere. So the sarcomere is going to be our functional unit of a muscle. So a muscle fiber is a muscle cell, but a sarcomere is the functional unit. And so this is where muscle contraction occurs. So at the microscopic, kind of all the way zoomed in, the sarcomere contracts. Millions of sarcomeres throughout a muscle all contract and shorten together when we think of a contraction. So as I just said, our sarcomeres are basic structural and functional unit of our muscle cell. So sarcomeres organized end to end from one another are what create our myofibril. And then Z discs are protein fibers that kind of serve as our anchor or border for each sarcomere. So I'll point that out when we get to our diagram here in a second. So one single sarcomere is going to extend from Z disc to Z disc. That's how we keep track of them. So remember, our thin and thick filament organization is what causes the striated appearance of skeletal and cardiac muscle. We call our contraction theory the sliding filament theory because our two sliding filaments, the actin and myosin, slide past each other, which causes sarcomeres to shorten. So as I said, I'm not going to really test you on I bands and A bands, but this slide kind of just fills you in on what they are. So our lighter bands are the actin, our thin filaments. We call these I bands. Our darker bands are known as A bands, and they are the length of our myosin or thick myofilaments. And our actin and myosin do overlap somewhat in our A band. And at this overlap, that's where our contraction actually occurs. So our actin is actually made up of three different components. We have actin, troponin, and tropomyosin. So our troponin has a binding site for calcium, and tropomyosin actually blocks where our myosin or thick filament can bind to our thin actin filament. Our myosin looks almost like a golf club in terms of having a head and a tail to it. And so in our myosin head, this is where they grab onto the actin. And then ATP, or our energy, has a binding site on our myosin as well. So let's take a look at this. So we're super zoomed in now. So let me zoom in. So remember I said a sarcomere is our functional unit of the muscle. This kind of zigzag line here is our anchor. That's our Z disc. So Z disc to Z disc is a sarcomere. So this unit here, from blue zigzag line to blue zigzag line, is one sarcomere. Our I bands have only our actin filaments. Our A bands have the myosin and where our actin and myosin overlap. The M line, you can think as the middle line, that's pretty much dead center. And so you see our thick filaments here in green have these little heads to them. So that's why they say they look like a golf club. And those are going to bind to actin. They're going to pull towards the M line when activated. And that's what is going to cause our muscles to shorten. So here's our myosin molecule. So you have the golf club shape where the head can grab on. So here in the actin, what happens is calcium is released. So let me zoom back up for a second. So troponin molecules have binding sites for calcium. So there is going to be calcium that attaches to troponin. When the calcium attaches to the troponin, it is going to help pull the tropomyosin out of the way. That reveals the binding site, and that way our myosin can grab onto the actin for a contraction. And when the myosin and actin contract and slide, we call that a power stroke. So for all this to happen, the first thing that has to happen is that our muscle fiber is activated by a neuron. So a motor neuron is a nerve cell that stimulates a muscle cell or stimulates muscle fibers. And the neuromuscular junction is the synapse or the connection between that neuron and the muscle fiber. A synapse is a cell-to-cell junction. And so it's either going to be with a nerve cell and another nerve cell, or a nerve cell and an effector cell, or the muscle fiber in this case. And then the term motor unit actually refers to a group of muscle fibers that one motor neuron stimulates. So sometimes we have really large motor units where one motor neuron stimulates thousands of muscle fibers. And we also have small, precise motor units where one motor neuron may only stimulate a few muscle fibers. Your lab for chapter 7 walks through this aspect really well also, so really pay attention to your lab this week because it covers a lot of our lecture material. So the neuromuscular junction structure. So you have your presynaptic terminal. This is going to be the end of a neuron. So The axon of a neuron is the part of a neuron that is going to be sending the signal. So the presynaptic terminal is the end of that cell axon, that neuron cell axon. The synaptic cleft, the word cleft means space. So this is going to be the space between our presynaptic terminal and our postsynaptic membrane. In our chapter here, our postsynaptic membrane is going to be our muscle fiber membrane. And remember, we call that the sarcolemma. So the cleft is the space between the end of our neuron axon and our muscle fiber sarcolemma. Synaptic vesicles are going to be located in our presynaptic terminal. They're going to store and release neurotransmitters. That's the way our neurons signal one another via chemicals. So in our muscle cells, this neurotransmitter is going to be acetylcholine, which you'll see is abbreviated ACH. So our presynaptic terminal is going to have vesicles of acetylcholine that it will release across the synaptic cleft to our muscle fiber sarcolemma. So we just talked about neurotransmitters. So they can either be stimulatory or inhibitory. And in terms of muscles, we're going to have acetylcholine as our neurotransmitter that stimulates our muscle cells. So here's just a diagram that illustrates that. So this is our axon of our neuron. It's going to have multiple synapses here with our muscle fiber. So if we zoom in even further on just one of these feet here, we have our presynaptic terminal. So this is going to be the end of our axon. We have synaptic vesicles filled with acetylcholine in this case. We have our sarcolemma, or our muscle fiber cell membrane. That's going to be our postsynaptic membrane. And then we have the space in between the two that is known as our synaptic cleft. As I said before, myosin and actin slide past each other during a muscle contraction, which is why we call this the sliding filament theory. So we talk about this in a very zoomed in version in terms of the sarcomeres shortening, but this has a domino effect. So when our sarcomeres shorten, our myofibrils shorten, which cause the muscle fibers to shorten, which cause the muscle fascicles to shorten, which cause our entire muscle to shorten. So this happens all the way at the microscopic level, but when it happens all at once and together, our entire muscle ends up shortening and contracting. So once our muscle actually relaxes, then our sarcomeres will return to their resting length. So for muscles to contract, we need energy. And just like our other cellular processes, this is going to come in the form of ATP. So energy is released when ATP releases one of its phosphate groups. So when you break that bond, energy is released. So ATP turns to ADP plus its extra phosphate group, and that is going to release energy that is stored in our myosin head. So in order for our bond or cross bridge between the myosin and actin to release, a new ATP has to bind onto the myosin head. So if you've ever heard of the term rigor mortis, this is when a person dies and the body goes all the way stiffened. And that's because no ATP is being produced any longer. So those cross bridges, or where the myosin and actin are binding, cannot be released. This is just a little diagram that talks you through that same concept. All right, we mentioned muscle tone when we were talking about muscle function. So remember, even at rest, in terms of maintaining posture, we always have some sort of muscle tone that is being produced. So this is the constant tension produced by our body over long periods of time. It keeps our back upright, it keeps our legs straight when we're standing, it keeps our head from drooping down and our abdomen from kind of just poking out. So this is going to depend on a very small percentage of our motor units. So one other concept that I guess we should touch on is the fact that when you decide or choose to activate a muscle, you don't have to necessarily activate it at 100%. So think about gripping something. If you're going to grip something heavy, you're going to squeeze really hard. You're going to activate a lot of your motor unit and as much of the muscle as you can. If you're just going to shake someone's hand, you don't need to squeeze as hard as you can. You're only going to activate a few of the motor units and muscle fibers within that muscle. So for muscle tone in our resting posture, we're really only depending on a small percentage of our motor units. All right, circling back to smooth muscle. Remember, this is our only type of muscle that's non-striated. And unlike our skeletal and cardiac muscle, we're usually only going to find one nucleus per muscle fiber. Our myofilaments are not organized into sarcomeres, so that's why it doesn't have the striated appearance, and they're involuntarily controlled. So they're pretty much any organ that's involuntary besides the heart. So our smooth muscle is under control of our autonomic nervous system, but it responds to neurotransmitters, hormones, and other internal factors. Okay, so cardiac muscles are striated. They tend to be quite long, and they also only have one nucleus per cell, unlike our skeletal muscles. So cardiac muscle contraction is autorhythmic, and so what that really means is that it has a set pace. So your SA node, your sinoatrial node, is the primary pacemaker for your heart. So it beats 60 to 100 beats per minute. So every second or a little more often than that, it's going to signal an electrical stimulus that causes our heart or cardiac muscle to contract, and that's going to happen like clockwork. So that's autorhythmic. So our cardiac muscle has a couple of unique features. One of these is intercalated discs. So they have kind of in between the different cardiac muscles, these discs that help make the connections really tight. It's important for our cardiac muscles to all be in sync with one another. So this is how we help ensure that when the signal to beat kind of gets sent, that all of our cardiac muscle receives the signal. Our cardiac muscles also have branches that reach out to one another, and this helps that electrical signal pass more simultaneously. So in terms of skeletal muscle anatomy, a couple other terms that you should know is the muscle belly. So our origin is going to be one portion or the attachment of the muscle that typically does not move. And then our insertion is going to be the other attachment for our muscle. So typically when a muscle contracts, the insertion is going to move towards the origin as the muscle shortens. But the bulk of the muscle or the mid portion of our muscle is known as our muscle belly. We have multiple muscles that work together. We call these agonists. One example of this would be at the elbow. So for example, our biceps and our brachialis both help flex or bend our elbow. So those would be agonists. The muscle or group of muscles that oppose a muscle action are antagonists. So in that same example, our triceps, which straightens out our elbow, would be the antagonist. So the agonists are what help with the motion. The antagonists are what do the opposite motion or oppose that action. So here's our example for our biceps. The origin is up here on the coracoid process of the scapula, and then it inserts here on the radial tuberosity. So as we contract our biceps, we cause elbow flexion or bending of the elbow. The insertion on the radial tuberosity is going to move towards the origin. Our triceps, which straightens the elbow, is going to be our antagonist. And then if we had our brachialis or brachioradialis here, a couple of other elbow flexors, those would be our agonists. So agonists help, antagonists oppose. Okay, muscles are named in a variety of ways. So that's really what you need to know is the fact that we have a multitude of ways that we decide on a muscle's name. So they're named for location, like the pectoralis muscle, which is located in our chest or pectoral region. The size, in terms of being a large muscle, small muscle, short or long, can determine the name. So one example of this is the adductor longus. The shape of a muscle, we have our pronator quadratus is an example of that. So that can help determine the name. The orientation of a muscle, for example, your obliques in your abdomen run at an angle while your rectus abdominis runs straight up and down. They can be named for the origin and insertion. For example, your sternocleidomastoid or SCM is basically named for the 3 different areas that it attaches. It can be named for the number of heads. For example, our biceps, bi meaning 2, has 2 different heads, while our triceps has 3 different heads. And then sometimes they're named for their functions, such as the abductor group, adductor group. So sometimes we just name them for their action. Okay, so you do not need to memorize all of the skeletal muscles. I'll highlight a few of them, and then on your study outline, I will make sure I include the ones that you need to know for the test. So we're going to kind of skip over the head and neck. This is just nice to know information. So these are all the muscles that contribute to turning our neck and moving our neck, as well as a lot of facial expressions. The one muscle that I want to point out is our sternocleidomastoid. This is a relatively large muscle that runs from our collarbone all the way up to the mastoid process behind our ear. And so if you bring your chin to your chest, you can kind of see your sternocleidomastoid or SCM muscle pop off of your neck. So that's a neck flexor. It brings your chin to your chest and also turns our neck back and forth. So that's a large muscle here on the neck, kind of running from the back to front. Within the abdomen, I'll point out your rectus abdominis here. This is your largest abdominal muscle, and it's kind of your 6-pack or 8-pack muscle. So that's your rectus abdominis. Some of the muscles in the back of the shoulder, we have our trapezius. So this shrugs your shoulders up. You also have a middle and lower portion of your trapezius, which help pull your shoulder blades together. Your latissimus dorsi is quite a large muscle here. You see it runs from this interior portion of the arm where its insertion is to this broad attachment in the back. So remember, a tendon connects muscle to bone. So when we have quite a broad attachment like we do with the latissimus dorsi, this attachment is termed an aponeurosis. An aponeurosis. So same thing with our abdominals. This broad attachment area would be an aponeurosis. You can see these small muscles here. These are our rotator cuff muscles. So it's cuff, C-U-F-F. You do not have a rotator cup. It's a rotator cuff. So these are shoulder stabilizers here. Some big arm muscles to point out. The main one I want to point out here is your deltoid. So that's the prime mover of your shoulder. So if you're looking at the shoulder from the front, the side, like we are here, or the back, you're going to have a portion of the deltoid. So that's going to lift your arm up. It's going to raise your arm to the side like a jumping jack. If you're rowing your arm back, your posterior deltoid is going to work. Another big muscle in our prime mover for our push in the chest is our pectoralis major. So a big chest muscle here is the pectoralis major. That's another one that you should know. Our triceps is on the posterior portion of the arm. It's going to straighten our elbow out. The biceps is on the anterior part of our arm. It's going to bend our elbow or flex our elbow. The brachialis is also going to help flex the elbow, and the brachioradialis is your drinking muscle. It also helps flex the elbow. So these three are all agonists in elbow flexion. Don't worry much about the muscles of the forearm. There's really nothing kind of important enough here to point out specifically for this class. Moving down to the legs, we'll talk about this as we go through it. So our gluteus maximus, that's your big glute muscle on the back. You also have a gluteus medius and minimus that are more on the side of your hip and deep to the gluteus maximus. So your gluteus maximus is kind of your power muscle for the hip, while your gluteus medius and minimus is more of a stabilizer muscle for the hip. This is showing the anterior thigh. So your quadriceps group is the most important group here. It's called your quadriceps because it's made up of 4 muscles. You have your rectus femoris, which is the only portion of the quad that crosses the hip joint. You then have your vastus medialis, lateralis, and intermedius. So quad means 4 muscles. Your quads are your most powerful muscle group. Moving to the bottom of the picture, we have our hamstring muscles. So you have 2 that run on the medial side, your semitendinosus and semimembranosus, and then your biceps femoris runs on the lateral side. So these all attach up to the ischial tuberosity of your pelvis, your sit bone. So your hamstrings are going to bend your knee, your quads are going to straighten your knee. Lower down in the leg, you have your gastrocnemius, which is your calf muscle. It does the gas pedal motion. You also have your soleus that does that gas pedal motion when your knee is bent. On the front half, the biggest muscle is your tibialis anterior. It's the muscle that lays right on your shin, and it helps pull your toes and ankle towards you. So you should know these 3 muscles contribute to aiding in breathing. So the external intercostals, the internal intercostals, and the diaphragm are all important for breathing. So this shows your intercostals are between your ribs. So the internal intercostals depress the ribs as you breathe out, while the external intercostals elevate. So think E external, E elevate. And that is the last for our muscle chapter. Like I said, don't spend your time learning every single muscle in the body. Take a look at your study outline to kind of help zoom your focus in on what may show up on the test. And then in the first half of the lecture, make sure you pay attention to all that kind of anatomy and physiology concepts.